Okay, welcome to this review of Chapter 15. This is going to be on the rest of the chromosomal theory of inheritance, which was briefly talked about in Chapter 14. And we're also going to cover pedigrees and some genetic disorders that are inherited through nondisjunction and a few other ways. So let's get started here. The first thing I want to talk about are pedigrees and how actual traits are inherited. Pedigrees can be used, again, to trace a history of a genetic disorder or a genetic trait through generations in a family. So through genetic counseling and things like that, actual jobs that are pretty high in demand right now, people can go and get their pedigrees traced to find out what type of genetic disorders exist in their family and how often they come about, often leading to what type of trait it is, whether it's dominant, recessive, or sex-linked. So to go through that, let's go through a few examples here. The first example I got is of a dominantly inherited trait. Now, when you're actually going through a pedigree, I feel that the best way to actually start categorizing whether what trait is what is by starting at the bottom. So with a dominantly inherited trait, we know that somebody with a person with a dominant allele, so one that is shown over another if it's an incomplete dominance, is going to be the one that comes up. So an affected individual who is this shaded in person is going to be, and actually this is a female because it's a circle, but this shaded in person is going to have to be possessing a capital A, let's say the trait that we're talking about, which we'll say is Huntington's disease. Okay, Huntington's disease is actually a dominantly inherited trait. So with Huntington's disease, anybody who has the dominant allele for that trait is going to get it. So this female right here has Huntington's disease, but since members of her family, and in particular her mom, didn't have it, that means she must have gotten one allele for the recessive trait. So this individual is actually heterozygous for that trait. Now, if we go up one, okay, and actually, if we go back down really quick here, the boy here must be recessive because that boy does not possess the trait. If we go to the second generation, we go up one, so we're working backwards here. This individual, the mom in the first example, must be recessive because she doesn't have Huntington's disease. However, the man, the father, must also be a heterozygote. Why must it be a heterozygote? Because his mom didn't have the trait either. Okay, so as these things get passed on through generations, they will pop up, but you can see that dominantly inherited traits actually appear more often than what we're going to see in the next example, which is recessively inherited traits. Okay, so the next one here, again, is recessively inherited traits. That can be anything from, Tay-Sachs to like hemophilia, things like that. And what that is, if we want to use B for an example, B, let's say, is hemophilia, and the little b is normal. In a parental genotype here, these two parents might be actually, hold on a second. I mixed those up. Sorry about that. So hemophilia is going to be the little b. Normal is going to be the big B. Okay. In the parental genotype here, both of these parents are going to be normal parents. But again, let's start from the bottom. Okay. Let's not always work from the top. Let's work from the bottom here. So this individual has hemophilia. So it's little b, making this individual positive for hemophilia. Now, in order to get that, it has to get it from somewhere. Okay, so actually, what is shown here is it's pretty obvious that this individual got at least one allele from his mom that was positive for hemophilia, this one. But the dad must have been a carrier for that trait because even though he didn't show hemophilia, he still passed on the recessive allele, ending up with one of his daughters having hemophilia here. Again, another instance of this would be something like, in this example, if we want to say that this individual has the disorder and its parents were the carriers of it, okay, we can just assume that even though they don't express that trait, that they're still passing on this gene. So whether, whatever disorder it is, and actually, I'm pretty sure, why don't, instead of using hemophilia, we use cystic fibrosis? That's a little better example here. Sorry, it's late in the day. So, no matter what the trait is, if it's a recessively inherited trait and it's getting passed down into a offspring, the parents have to have at least been a carrier of the gene. Whether they're a carrier or they actually express that trait, it doesn't really matter, because the odds of passing it on to their offspring is still that 50-50 chance in this example, or in this example. So there's a 1/2 chance that the offspring is going to be 50 with cystic fibrosis, and with this example, the chance is going to be 1/4. So that is for recessively inherited traits. And now with sex-linked traits, let's do red-green color blindness. In this example, if we start from the bottom, the female here, you can tell, is a carrier for color blindness, because she is actually possessing 1/2 of her X chromosomes have the color blindness gene on them, and the other half is fine. And with sex-linked traits are located on that X chromosome. So again, when females carry that X chromosome, they have two options to have the correct or the wild type gene. Males actually only have one, because they only get one X chromosome. So with this male here, he has the wild type X chromosome, and he also has the Y chromosome, making him not colorblind. Now, however, in this example, if we look up here with these parents, the father had XY normal, and this mother had a normal X but was a carrier for color blindness. The genotype for both of these individuals here is going to be Y from dad, but they're both going to get this chromosome for color blindness. So they end up both being colorblind. So that's just kind of a little overview on how traits like that are passed down through generations. Next, we're going to talk about gene mapping. And one of the things I wanted to show you before I get there is just kind of a cross between some of the very famous flies that we talked about in our lab. Some of the GAs in Thomas Hunt Morgan's lab actually went through this kind of experiment and figured out a way on how to map out genes on an actual chromosome. So what they did was they took the parental phenotypes of a certain fly, okay, and the phenotypes for these flies are gray and normal winged for females, and black and vestigial wings for males. And when they mated those together, they would have expected, since the trait is wild type, recessive, wild type mutant, and that's mated with all mutant, you would expect to get a 1:1:1:1 ratio. So you'd have an equal amount of offspring for each of the 4 categories. So that is represented by these numbers down here. The expected is not actually what they observed. What they actually observed, out of all of the offspring, they observed these ratios instead. So there were a lot more individuals resembling the parental genotype, which are these two, black, vestigial, and wild type, because those resemble the parents. And there were a lot less of the recombinant flies, or the flies that were mixed traits, so ones that had vestigial wings and gray body, okay, or the other way around. And they figured out that that was because that law of independent assortment didn't always hold true for chromosomes on any particular gene. And it turns out that both of these genes were located on the same chromosome. So how does that happen? Well, what they did was they took chromosomes from the female fly, and through crossing over, if crossing over occurs where the chromosomes lay on top of each other in between the 2 particular genes you're looking at, what it's going to do is create a recombinant DNA or recombinant gene there. And the recombinant chromosome has the one or wild type allele for gray body, or on the other side, it has that vestigial wing. And that doesn't match any of the parents. But the odds of that happening are actually a lot less than the parental phenotypes, which are if the crossing over happens outside of this gene map. Okay, so as these two are mated together, they look at the crosses at the end and they figure out that the recombinants come out a lot less often than the parental phenotypes, and they actually used this data. They added these two up, got 391, and used that data to compare to the total number of offspring, getting a 17% chance that the chromosomes crossed over. And because of that, they were able to map out on a gene what that looked like. So actually, let's back up a little bit before I show you the map. So 17% of the time, black and vestigial traits crossed over. One other trait they were looking at were cinnabar eyes, and cinnabar eyes are just kind of like a lightly brown shaded eye. That crossed over with black body 9% of the time, and cinnabar eyes and vestigial wings crossed over 9.5% of the time. So they looked at all three of those traits and figured out the percentages of how often they crossed over. And when they looked at the chromosome and actually mapped it out, they could eventually figure out that black was 17% likely to cross over with vestigial wing. So that meant that it was 17 genetic map units apart. Now, that's kind of an artificial way of measuring, but it's just kind of what those scientists developed in a way to figure out how to actually map out multiple genes on a longer chromosome. So if you're ever looking at a chromosome like this, you can always substitute the percent for GMU, or genetic map unit, and those genetic map units then could be used to not only map these three genes out, but map a ton of other genes out that lay on the same chromosome. So that was a really huge discovery in genetics, and we've actually now mapped the entire human genome out, so it is possible to see all these individual genes now on every chromosome. Last thing we're going to talk about are a few genetic disorders. So with regards to genetic disorders, we got a few kinds. We got recessively inherited, dominantly inherited, and disorders located on the sex chromosome. And then we got some disorders located received through the process of nondisjunction. So we'll go through each one of those. Some recessively inherited traits are cystic fibrosis, Tay-Sachs disease, where you actually collect a lot of lipids and you're not able to function or metabolize those lipids, and then those lipids actually end up killing you pretty young when in age. So, and then sickle cell anemia is also another recessively inherited trait. However, this trait is pleiotropic. I hope I spelled that right. No, I did not. Tropic, okay? And being pleiotropic, it has a lot of different effects. It actually has a few different effects on an individual who has it. So if you are dominant for sickle cell anemia, you have the trait and you're probably going to die. Or I'm sorry, if you're homozygous. So if you're homozygous for sickle cell, you have it and you're going to die. If you have sickle cell, if you're a sickle cell carrier, but you have normal cells, you are going to, I kind of wrote that the wrong way, normal cells, sickle cell anemia, you're not going to actually receive any sickle cell symptoms, but you are going to be resistant to malaria. So that's a huge benefit to people who are carriers of sickle cell anemia. And somebody who has normal blood cells, you know, they're not going to have any symptoms of sickle cell anemia, but there will be receptive to malaria. So there's almost a heterozygote advantage to having sickle cell anemia. Lastly, on this slide here, we already kind of talked about dominantly inherited traits like Huntington's disease. What Huntington's disease is, it affects your nervous system. You start losing coordination, and you end up actually dying by the age of maybe 30, 35-ish, but you don't find out you have it until later in life. So passing it on is actually pretty common in lineages and through generations because people have already had kids by the time they figure out if they have Huntington's disease or not. Some sex-linked traits to go along with this. Sex-linked traits like eye color or hemophilia or color blindness, they are all located on the X chromosome. Males have a lot higher chance of getting these sex-linked traits because they only have one X chromosome. Females get two X chromosomes, so the odds of them actually receiving a trait or a mutant allele on the X chromosome and expressing it is kind of low. So who is responsible for these inheritance of sex chromosomes? Well, it's the mother, because the mother is always passing on the X chromosome, and if a boy receives that X chromosome from his mother and a Y from his father, then it's the mother who has given that condition to the son. And most of the time it's because that mother is a carrier of the trait and doesn't express it, yet they pass on that that gene that they've carried to the son, and now he has no choice but to express that gene, so they end up with the disorder. So hemophilia, the inability to produce platelets in your blood and clot your blood. It's not as dangerous nowadays because they have synthetic platelets that you can take, so not as dangerous anymore, but was dangerous way back when. Red-green color blindness, which is also inherited on the X chromosome, really common actually in populations of males, but the color blindness gene is not really that detrimental. You just can't fly fighter jets or, join the Air Force, I guess. Oh, and you can also be an astronaut, which is pretty sad if you have color blindness, but don't worry. If you want to be an astronaut, there's still hope for you, okay? And then with muscular dystrophy, that's also caused by sex-linked inheritance. So those three are what we're talking about. Last thing here we're going to talk about is nondisjunction and some traits inherited through that. Nondisjunction is really a process where microtubules later in life, after fertilization, actually don't work properly, so they end up not splitting up the chromosomes properly during either meiosis 1 or meiosis 2. If it occurs during meiosis 1, the chromosomes are going to be messed up the whole process, or the whole way through, because these two chromosomes have now traveled into the daughter cell of the next level, and then as they go through meiosis 2, they've gained one extra chromosome, where these have lost one chromosome. So through that entire process, every single daughter cell in that whole way down is going to be affected by this nondisjunction. If it actually happens in meiosis 2, however, the effects are seen less, because it's already happened normally here, so this cell has the correct number of chromosomes and it's going to end up with the proper set of haploid DNA. However, if it happens in meiosis 2 like it does over here, then one of the eggs is going to end up with one extra chromosome and the other egg is going to be missing one. And that's going to be a problem if those eggs end up being used to fertilize and come up with a new organism. So some of those, any change in chromosome number, which I just showed you, is known as aneuploidy. Aneuploidy is that general term, just any change in a chromosome number. Trisomy is dealt with when you have an extra chromosome of one type. So if you have an extra chromosome 21, you end up with Down syndrome, which means you have slower development, and you live it's not always common that you live a shorter life, but it's actually pretty common, and you have trouble learning new things at a faster pace. So Down syndrome, you got Edwards syndrome, Pateau syndrome. Those are some of the symptoms or syndromes you can actually live with, but they're pretty detrimental to somebody's development. And then sex chromosomes here, the only one that you can actually survive with that's monosomy is Turner syndrome, where you get one X and you don't get any other sex chromosomes. The other ones deal with extra chromosomes, so Klinefelters, where you get extra X chromosome. Both of these, you have underdeveloped sex organs, and you end up dying, I think, pretty young. So, and then super female and super male, you actually can't tell if somebody is a super male. They're just really tall. And with super females, you wouldn't really notice a difference with them either. They just have that extra X chromosome. The only way to find out if you are a super female is to get genetically tested for it. So that's going to be the wrap up of chapter 15. Hope this helps. And good luck on the test. If you have any other questions, let your teacher know, and we will see you next time. Thanks for listening.